Hello, my name is Natalie Castell. I'm a lab animal medicine resident as well as currently a PhD student here at Johns Hopkins. I'm happy to share with you today some of our data on socially housing our SIV-infected pigtail macaques. So social housing, I think everyone here understands, is very important for non-human primates. In addition to this, non-human primates are a really important resource for understanding human infectious diseases, and so these guys represent a really vital lab animal for this research. Pair or group housing of social species such as non-human primates is considered the standard per the many regulations that cover research animals, including the guide as pictured here. So as I mentioned, social housing is very important for animal welfare, but there are some study goals that can sometimes preclude social housing, especially in infectious disease studies, and these can include transmission of the agents, the ability of researchers to access the animals, the ability of animals on a study to have surgeries or implants that remain intact and not accessed by the animals, as well as concerns regarding the integrity of study data when animals are able to interact directly with each other. And there's a lot of really good literature out there already showing that single housing acts as a really important psychosocial stressor, specifically in the context of macaques and non-human primates. So I chose a few studies here to highlight examples of that. There's some great research showing both that social housing is an important social buffering and coping mechanism for these animals, as well as various ways that this affects their physiology and specifically can directly alter their immune responses. So our lab uses pigtail macaques as a unique model that recapitulates a lot of the central nervous system pathology that occurs in people infected with HIV. We inoculate them with this combination of SIV viruses listed here, and most of our studies utilize juvenile to adult male pigtail macaques, and they all pretty much receive the same treatment for the first few weeks after infection before they start different therapies depending on what study they're a part of. So in 2012, after the release of the latest edition of the guide, we revisited whether our animals truly needed to be singly housed for these studies and made a transition from all the animals being housed individually during infection to keeping them in compatible pairs or trios throughout the entire duration of the study. And because of this body of evidence that I already mentioned about the effects of social housing on immunity and infectious disease, we looked at the data from grouped studies and assessed how this has changed outcomes of infection. And what we saw was some pretty striking differences. So just to start off with, acute infection data of those first 2 weeks, where animals can all be grouped together, showed that the animals prior to 2012 that were singly housed had significantly higher both blood and central nervous system levels of virus. And so we were really interested in what immune parameters might be altered to lead to this kind of big change in outcome. And what we saw is that T-cell responses were similarly different between the housing groups. T-cells are really important for SIV and HIV infection because they're the main cell that is infected by the virus, and they're also a critical cell in our adaptive immune system. So they're both responsible for being infected and for fighting the virus. And what we saw was that in the singly housed animals, there was a greater decline in both of their major types of T cells, so both CD4 and CD8 T cells during that acute infection period. And we also looked at the activation of these cells in the bottom two graphs here. And what you can see is that there's much greater activation of both types of T cells at 1 week after infection. And while activation is associated with, an immune response, because these cells are infected by virus, their being activated also allows for greater viral replication. And we also looked at changes in the innate immune system. So specifically, these graphs are showing circulating monocyte levels during that same acute infection period. Monocytes are really important for SIV infection because they're associated with bringing the virus into the brain. And what we saw was that, again, at this 7-day post-infection time point, there's a difference between groups, with the socially housed animals having more monocytes. This is mostly driven by this classical subset of monocytes that are CD14 positive. And I won't get too much into the immunology of things, but CD14 monocytes are considered to be resistant to infection, whereas this other subset that's also increased, that are CD14 positive, CD16 positive, are known as intermediate monocytes, and these are the ones thought to be important for bringing virus into the brain. So it's interesting that both subsets are increased in the socially housed animals. And similarly, there are differences in platelet activation between these animals. So platelets actually act as a very important immune cell as well. And for all animals after infection, they experience a pretty dramatic decline in their platelet numbers. But we saw that the socially housed animals recovered back to their normal level much more quickly than the singly housed animals that still had reduced platelets 2 weeks after infection. And P-selectin, MHC1, and CD40 ligand are all markers we can look at on platelets to assess their activation status. And we saw that specifically for P-selectin and MHC1, the socially housed animals showed greater platelet activation responses. So I'll try to keep things very simple for brain metabolites because this is definitely not my area of expertise, but we used the cerebrospinal fluid samples we have from these animals to assess how different brain metabolites and neurotransmitters changed over the course of the first 2 weeks of infection in these groups of animals. And to start with, tryptophan is an amino acid that is the precursor to serotonin. But in the metabolic pathway for this amino acid, it can also be made into quinolinic acid. So these are kind of like differential outcomes for tryptophan. And we saw that in the singly housed animals, they had greater increases in tryptophan after infection, but this led to greater levels of quinolinic acid rather than increasing serotonin. And quinolinic acid is an NMDA agonist that has been associated with oxidative stress and inflammation. So kind of diverting into this pathway is broadly considered to be a negative outcome. And similarly, phenylalanine is another amino acid, and it's the precursor for dopamine, which is a neurotransmitter that is associated with cognition and learning, as well as motivation and reward. So broadly, dopamine is kind of considered a good or a happy neurotransmitter to have, but it's also the precursor to epinephrine, which has been shown in some of those papers that I had pictures of earlier to directly modulate immune responses, and in general having increased levels of dopamine and especially epinephrine can be associated with higher levels of anxiety and insomnia, at least in people. So we saw that the singly housed animals had higher phenylalanine as well as dopamine after infection compared to the socially housed groups. And more broadly, we just kind of looked globally at the standard deviations between the different immune metrics that we were able to look at in our animals, and found that the socially housed animals in general have lower standard deviations than those from the singly housed studies. And this suggests to us that social housing might be a kind of refinement to our research model in that it could improve data reliability and reproducibility. So one of the concerns about socially housing macaques that are SIV infected is that they could transmit virus between each other and either create a superinfection or somehow alter their course of disease. So we compared the viral sequences between 3 pairs of macaques using a number of different samples that we had from these animals. So we looked both in their central nervous system as well as their blood and different lymphoid tissues. And While there was pretty limited sequence diversity, as you can see from this graphic here, we were able to construct kind of little phylogenetic trees of the relationship of viruses between the animals, and you can see from this that there doesn't appear to be a significant association of the pairs with their viral sequences. So in summary, I've shown you a number of ways that pair housing our pigtail macaques has altered both their early adaptive and innate immune responses to SIV infection. And because these guys are able to interact with each other normally and benefit from social buffering, this may actually represent a more accurate representation of what occurs in people with HIV. In addition, we were able to show that overall our data variability was lowered in the pair-housed macaques, and that there isn't any evidence at this time that there is viral transmission occurring directly between the pairs or affecting the outcomes of this research. So the take-home points is that social housing of SIV-infected macaques represents an important refinement to our model of HIV pathogenesis. And I would like to thank everyone in the retrovirus lab and outside of the lab that helped us complete this research. There are far too many people to thank individually. And with that, I am happy to take any questions.